Let's talk about HIV entry. This is the process for how we are going to get the components of this little inner cone-shaped spheroid into the cytoplasm of the cell. And actually, we're not even going to really stop in the cytoplasm for this particular virus. We're going to dump out this entire core. The core is going to loosen up, travel to the nucleus, and then it's going to basically dock at the nucleus and dump the contents in there. One way or another, though, we need to make sure that we get what's inside of this to the inside of the cell. And so for a normal virus, not HIV, this would just involve fusing with one or more, actually just usually one, cellular membrane. Most commonly, this is going to be fusion with an endosome membrane. So endosomes, endosomes, are going to be little vesicles that the cell brings in from the surface and then brings down, like that. And there's a process, and there are a couple enzymes involved, and we will cover that a little bit later in the fusion mechanism because it's important. The virus basically is set up so that this thing can happen, and that when this does happen, that will trigger the next events in the fusion cascade. So we'll start out with attachment. The protein that's going to be responsible for attachment is the envelope glycoprotein, which is ENV. That's how we abbreviate it. In this particular image, they're calling GP120 the surface subunit, or SU. I still like to call it GP120. You can call it whichever makes you happy. The bottom of the protein, which is a series of long helices, I would call GP41. They're calling it the transmembrane part. I don't really like that name because only a little bit that's actually not shown here at the bottom is transmembrane. The rest of it is what we would call a fusion motor, or a class 1 viral fusion protein. We will study class 2 and 3 viral fusion proteins a little bit later. We'll just sort of mention them. But this is a class 1, and class 1s are the most common. The nice thing about this class 1 viral fusion protein is that once you understand how it works, you understand how SARS-CoV-2 works, you understand how influenza works, you understand how a lot of other viruses work as well. Class 1s are the most common ones. You find them in every coronavirus, I believe. Most enveloped RNA viruses are class 1. And then second most common would be class 2, and third most common would be class 3. All right. Oops, go back. What you will see with viral attachment and fusion proteins. So the bottom part, GP41, is going to be the fusion protein. The top is going to be more of an attachment protein. In some other viruses, they'll actually split up this function. They'll have one whole protein anchored to the membrane that's just doing attachment, and another one that is just doing fusion, but is going to be linked to the one that does attachment, and so they're going to help each other. It's just basically the difference is whether you have two little transmembrane regions, one for each subunit, or if you only have one transmembrane region for the entire thing. For HIV, there's only one transmembrane region. It's down at the bottom of GP41, and so that is going to anchor the entire complex of GP120 and GP41, the whole of which is called the envelope glycoprotein, ENV. What you will see with a lot of these attachment and fusion proteins is that they're going to bind multiple receptors. The first receptor is usually going to be something very generic or something very tall that sticks up very far outside of the cell membrane. This is just going to be to catch the virus onto a potential cell where it could then bind to another protein and actually start to go through the motions of fusion. What you're missing in this diagram over here is extracellular matrix. The outside of the cell actually has a pretty thick layer. Like imagine a layer going to about here, with only just the tops of these long proteins poking out from that. The other proteins are there, but they are down in. It's like being under a forest canopy, kind of. And there are a lot of different glycans and proteins and all sorts of stuff you'd find outside of the cell as part of this extracellular matrix. So while a virus can bind to something in the extracellular matrix, that's usually not going to be the main fusion receptor. That would just be an attachment receptor. And receptor is the word that we're going to give to the cellular protein, the host protein that the virus is binding. If the virus sticks to it, it's a receptor now, whether it wanted to be or not. There is no consent in virology. Yeah, isn't that great? Yeah. Okay, so our handsy little virus is going to grab onto one of these long proteins, and then that gets it attached to the cell, and it can then over time work its way down and eventually arrive at another protein that's going to be closer to the membrane. So usually it is this two-step process. For HIV, the protein that's going to bind first is going to be any one of these things. CD4 is a common one. This is structured like an immunoglobulin in that it has immunoglobulin-like folds. Immunoglobulin is a fancy name for antibody. So it's made of the same sort of shape of Lego bricks, if you like, as an immunoglobulin. But this is a protein that's found on the outside of T cells. I think almost exclusively. There are a couple weird cells in the gut, but those might even be classified as T cells. So you would have CD4. It's a co-receptor that T cells would use. DC sign and L sign, these are both C-type lectins. Lectins are for grabbing onto, I believe, sugars. And HIV can also stick to some degree to heparan sulfate, which is going to be found on a lot of different proteins on the surface of glycoproteins. And it could bind to glycosphingolipids, which is a lipid, so it's going to be right down here, but with a little glycan chain. Now, those are probably not long enough to serve as a meaningful primary receptor, only because they would be buried right down in the extracellular matrix. And a virus doesn't have a way to sort of, motor itself through the extracellular matrix. It has to catch on something that's sticking out, and then once it's caught there, then it can just sort of, get blown around in the current, bloodstream, airstream, whatever you think. Actually, enteric, it would be the poo stream that it could also get caught in. That's fine. That would give it some motion, and eventually it would bump into its actual entry receptor. So the attachment receptor is going to be necessary and sufficient for the virus sticking to the cell. It will be probably transient in that if the virus sticks to the cell and doesn't enter after a certain amount of time, eventually it'll come off. It's a normal reaction with a K on and a K off, like that. Just like you see in chemistry all the time. The entry receptor is going to be the one that actually catalyzes virus membrane fusion. And the membrane fusion process for this kind of class 1 viral fusion protein, that is going to be irreversible. It's going to be a set of changes that take us from a high energy state stepwise down to lower energy states. And because we're losing free energy, if you want to think of it as that, at each step, there is no way to go back up unless there's a way to put energy into the system, and there is not a way to put energy back into the system. So thinking of it from the virus's point of view, it's got one shot, just like the Eminem song, this is its chance, so yeah. Better flow, right? Yeah. Okay, that was stupid. Anyway, here we go. Here are the structures of some important proteins that are going to be involved in the entry process. The attachment receptor is going to be CD4. That's shown down here. This is the top of CD4. It does have a transmembrane region, but the transmembrane regions on a lot of these proteins are very difficult to solve as part of an X-ray crystal structure. So often you will see the protein cut off, and you'll have to just imagine, like, we know there are transmembranes there. We just couldn't solve the structure of them. And people don't like to just randomly predict the structure. They would rather actually show it because sometimes it's not exactly what you would have predicted. So there is CD4. It has 4 immunoglobulin-like domains, and HIV is specifically going to grab onto domain 1, which is the one on the outside, the very tip domain. And then the transmembrane would be coming off of domain 4 of CD4. All right, so that's going to be the attachment receptor. The thing binding it is going to be HIV-1. So it says here GP160, but you know and I know that's just a code for the envelope glycoprotein, both GP120 and GP41. And you know that the math doesn't quite add up, but yeah, it's close enough for protein gel. And so there it is. There is our HIV envelope glycoprotein. And we've cut off the transmembranes to solve the structure. They actually exist in the real world. All right, there are going to be several actual entry receptors. Now, these are the ones that are going to cause the irreversible changes to start. So those are going to be CCR5. That's the most common first receptor. CCR5 is going to be common on macrophages, if I remember correctly. And then CXCR4, this is going to be common on T cells. So the early virus that comes in tends, like when you're first infected, it tends to be CCR5 specific. And the later virus that evolves inside of a person as it's sort of spreading out and colonizing more spaces tends to shift to bind to CXCR4. These are both chemokine receptors. They're named after a motif. At the center, it either has 2 cysteine amino acids right next to each other, CC, or it has 2 that are separated by 1 C. The X means something, and C. There you go. And there are a bunch of them, and there is a ligand, a natural protein that is supposed to bind to each of these. And so normally you'll see that called CCL5. CC chemokine receptor ligand for number 5, as opposed to CCR5. And so CCL5 will bind CCR5, CXCL4 will bind CXCR4. Yeah, ligand receptor, that's all it is. And that's just naming, and honestly, you don't have to know why they're called that. You can just remember that they are called that. These are 7 transmembrane G protein coupled receptors. You will have probably heard of those in other classes. G protein coupled receptors, people will call them GPCRs. They are super involved in cell signaling. Almost all the drugs that you would ever take in your life, you are taking to bind to a particular G protein coupled receptor. They are important for how we do things, basically. Yeah, big time signaling. All right, so we've introduced those. And now the last thing is going to be, here is our CCL5. So this is the CC chemokine receptor ligand, number 5. You don't have to spell that out. You can just put CCL5, and I'll know what you mean. It binds to CCR5. You probably can't see a big difference here, but you see how these helices are kind of splayed out? They're kind of tilted further away. And when the ligand binds, they get pulled back in. You get a conformational change on ligand binding, and it's like a bear trap. It sort of snaps down on the ligand to hold it into place. And so that's what's going to happen. This is the normal case. This is what CCR5 is for. This is what CCL5 is for. It's a little message. It comes along. CCR5 snaps down. When it snaps, that's actually going to move this bottom domain right here. And we're going to move the bottom domain, and it's basically going to swing down like in a Rube Goldberg machine, if you're familiar with those. You know, just like with all the contraptions where the boot swings over and kicks the handle of the toaster that makes toast that pops out and does the thing. You see them in Tom and Jerry cartoons. If not, it's not important. Just if that's in your brain, hang it on that peg. So this is going to swing down and actually bashes into, we think, the G protein, which is coupled to the G protein coupled receptor, and that's going to start the signaling cascade. In this case, the virus is trying to sort of intercept this process. It's trying to use this mechanism. The virus is going to stick a little loop at the top of the envelope protein down into that pocket. And darn it, if that loop doesn't do a pretty good CCL5 impression, and it's going to cause the same thing. The protein, the receptor, is going to snap a little bit on it, and once that locks our glycoprotein into position, then the next thing that happens is that basically when you catch one, you reel it in, right? Yeah, you know fishing. Yeah, or whatever. You've seen it on TV. Yeah. Same thing for the virus. The virus is going to make the cell think it's caught a little CCL5, and then once the cell catches one, it usually pulls in the receptor. So it's going to make an endosome around that receptor and bring it in. And unfortunately, it's going to bring in the HIV that's attached to it. Yeah, the cell doesn't know this, and so the cell gets fooled. Yeah. So you can't get fooled twice, but our cells get fooled all the time by viruses like HIV over and over. So I don't know. Yeah, somebody didn't know what they were talking about. So yeah, there is the envelope glycoprotein. It looks very nice, and it is going to be synthesized in the endoplasmic reticulum, and it is going to come out, and it's going to be on the surface of the virion. Now there's a little site between GP120 and GP41 that is going to be cleaved. That can happen at different times. So it's not going to happen immediately. Like when the glycoprotein gets folded up and glycosylated and 3 copies come together to make the trimer, because it is a trimer, 3 copies of the envelope protein, it's not going to be cleaved at that time. And the cleavage is necessary in order to activate fusion. So the cleavage has to happen, or else all the other things after can't happen. When does the cleavage happen? It probably varies. There are many proteins that would be associated with the cell. They'd be in that extracellular matrix, and they would be able to cleave the envelope protein. So in our case, it's going to be what we call a furin-like enzyme. That's F-U-R-I-N. And we'll show you the furin cleavage site and talk about that. There's actually 2 of them in this virus in just a minute. Okay. GP120 part of this is going to actually do the attachment to the CD4 protein. And it's going to attach at a very particular place, and we're going to show you on the next slide. And that's the kind of detail that gets you the good kind of points on exams. Just saying. Not telling you how to live your life. Just saying. So these blue things are the CD4 protein, and it's domain 1 at the tip of this that is actually going to bind into, it's a little tiny pocket on the glycoprotein on the top of GP120. Here is just GP120. So we took away GP41, it's thrown away for the purpose of this. And you can see there's a little pocket there, and it's got a little star in the pocket. So by pocket, I just mean a slightly dented in area. At the bottom of this pocket is going to be a phenylalanine, and it is going to be phenylalanine 43. There it is, right there. Boom. So PHE43 or F43, that would be a good way to say it. That phenylalanine is actually going to be the one that sticks to, is responsible for sticking to the CD4 domain 1. So CD4 is going to attach, and it's going to attach to a pocket, and at the bottom of the little pocket there's going to be a little phenylalanine. There we go. And it's amino acid number 43 of the glycoprotein. So that's why it's called phenylalanine 43. Great. These are, these are other possible attachment receptors, M48, U1, BNM3, 170, and they also are immunoglobulin-like, I believe, and they're going to bind at this same little pocket site. So that's the pocket you've got to hit, and when you hit that pocket, whenever you see an enzyme binding to a substrate, it's actually going to change shape. There's a thing called an induced fit. where the enzyme will usually be kind of open, and then when the substrate wanders in, it's going to bind to things, and that's actually going to close the enzyme around it. That changes the position of some parts, chains of the amino acids, and that's how the catalytic activity is actually going to happen. Yeah. So yeah, some kind of induced fit, and the induced fit is step 1 in our little energy curve. So we have a phenylalanine that's an unhappy phenylalanine, because phenylalanine is hydrophobic. Yeah. And this phenylalanine is a hydrophobic thing that's exposed to water. Oh my gosh, that's the one thing it fears, right? It doesn't actually fear it, it just repels it. Yeah. Only humans can feel fear. I guess animals probably too. The pocket, therefore, is at a higher free energy when it's around water, because the water is being repelled by it, so the free energy of the system is higher. Think of it that way. When you bind that part of the protein in, and I don't remember which amino acids actually participate, it may be multiple ones, I suspect they are hydrophobic ones, that then creates a slightly lower energy state. So we've gone down one little step. Congratulations. Oh, pardon me. These are 2 therapeutics that bind in there. They are not co-receptors. Sorry, I'm losing my tiny mind. But they're going to bind in there, and if they bind in there, that will then block binding of the GP120 to CD4. So that's a potential way to stop the infection before it happens. So that's pretty cool. Yeah. Sorry about that, and let's carry on. Okay. So that's the attachment step when we bind to CD4 or one of those other things, DC sign, L sign, for example. And I do not know the binding pockets. I'm not sure if there's a good structure to illustrate it. I believe all of the binding faces have been worked out for all those things. Anyhow, shown here at the top, this gray thing is going to be our CCR5 molecule. And so it's just shown sort of floating in space, and we've actually cut off the front of it. That's what this sort of uniform gray stuff is. It's actually going to be buried in the membrane. So the membrane is going to come up to right about here, and so there's just a little pocket. It's like a little pocket in the membrane, and CCL5 is going to work its way down, and there's an alpha helix in CCL5, and that alpha helix is actually going to bind into the groove of CCR5. So alpha helix of CCL5 would normally bind there, and it's going to make contacts. The side chains on this helix, and by helix I mean, look at this little curly bit there. That's the sign for this will be a helix. It's going to interact with the pocket. It's got amino acids to do that. The virus, GP120, has a region called the V3 loop. It's one of the variable loops that changes from virus to virus, and this is variable loop number 3. Variable loop number 3 folds up into a little alpha helix. The little alpha helix has side chains that are able to reach out, and darn it if they don't just fit perfectly and make nice little interactions with the pocket. So we're putting the same shape in there, and we're putting the same or similar side chains that make similar hydrogen bonds and into this environment. And so yeah, you just got a little helix, and it goes down into the pocket, and that is going to cause the same conformational changes. So you're going to get basically the CCR5 was like this, and it's going to boom, snap shut like that. And it's less dramatic than me clapping my fingers together. You move from the red one to the purple one. But yeah, that's the idea. When that happens, that will then activate CCR5 and activate its little G protein coupled receptor, which I don't think the virus particularly cares about. I think this is just a convenient thing for the virus to grab, but probably it has evolved to either deal with whatever happens or to actually love whatever happens with CCR5 activation. Yeah, you're welcome to look into that. That would be a nice little add-on for a test answer. Okay, GP120 is going to interact with the N-terminus. Actually, it interacts in the pocket, which is kind of a big part of CCR5. that is going to move the V3 loop of GP120 into position. So after it interacts with CD4 and bumps into the N terminus, which is out here of CCR5, that is then going to free up the V3 loop. I do not remember which part of the virus first bumps in. Yeah, I want to say it's V1, but I'm going to have to look that up, and maybe you can look that up. With the N terminus of CCR5. But then the V3 loop is the one that actually fits into the pocket of CCR5. It mimics the structure of CCL5, and it's sort of the same look and the same sort of hydrogen bonding level. And you can actually make a drug called Maraviroc, and it goes in there and it does all the same sort of hydrogen bonding to block the bottom of the pocket. So it's sitting in that little pocket of CCR5, and because it's already in there, HIV can't slip its grubby little V3 loop into the pocket. So yeah, good. Everybody wins, I think. Yeah. Maraviroc. It's not directly an antiviral because it doesn't directly bind any viral component, but it binds to its host component and it prevents the virus from using it. So yeah, it's kind of a taking something away from the virus that it desperately needs. Now, the downside of that is Maraviroc will prevent, I believe, CCL5 from binding. So if you need that CCL5 signal, you're not going to get it through. And so there would be some sort of problems with that. But overall, if I remember correctly, CCR5 is not critically important. If it gets messed up, knocked out, misfolded, people survive. People are walking around with deletions in that. I think something like 1 out of 5 of you out there probably have a deletion in this. It affects your ability to get long-term HIV and like progress all the way to AIDS. And it has relatively minor costs, slightly increased risk of heart disease, if I remember right. Okay, almost done with this section. Almost done. So now this should have been a little earlier, but here we have, this is the CD4, and here it is docked with GP120, so the colors are showing you what colors these things are. And so it's going to be docked in there, it's going to be binding to phenylalanine number 43. And when that happens, that is then going to pop out the V3 loop. The V3 loop is going to then fit down into the pocket, and there may be another interaction here with GP120 interacting with the N-terminal region. That's the interaction you can see right up there. Between those two things, you can think of them as 2 steps maybe, you will eventually get the V3 loop popped out because it was happier being inside, but now that it's out, it can bind into the pocket of CCR5, and that takes us down one more energy step because it's real happy in the pocket. Real happy. Real happy, you guys. All right. And so binding is going to change the structure of all of these things. We're going to change the structure of CCR5, definitely. Boom. We're going to change the structure of GP120 several times to like make it possible to bind the N-terminal domain, to make it possible for the V3 loop to be loose and flopping around to fit down in that pocket. It'll also slightly change the structure of CD4, not in the way that I think means anything, but yeah, whenever you have 2 things interacting, both of those things are going to change in structure, or I guess at least one of those is definitely going to change in structure. Yeah, that's just how interactions work. They move things. Okay. In the next section, we will talk about internalization. We will talk about pulling these in through clathrin-coated pits. But until then, thank you very much.